we go ahead and get started then, if you're ready?
Yeah.
Cool. Okay, so it's my pleasure to welcome Eva Dyer. She is here in Atlanta in the Department of Biomedical Engineering at Georgia Institute of Technology. She leads the NERDS lab, the Neural Data Science Lab, which is a cool name. And has a lot of different interests in intersection between neuroscience and machine learning, looking at approaches to interpret complex data sets, and also methods that are inspired by the organization and function of biological brains. So she has a background in electrical and computer engineering and comes previously from Rice University. And before that, University of Miami. So without further ado, Eva, I will let you take it away, and welcome to the Brain Space Initiative, and we're looking forward to your talk.
Great. Thanks so much, Vince and Armin, for organizing this and inviting me. Yeah, so as Vince was saying, my work focuses is at the interface of both machine learning and neuroscience. And today what I'll talk to you about is some work that we've been doing in being able to essentially form, low dimensional representations of data, and then think about how we might go about comparing different types of data sets through essentially forming representations and then aligning them in order to bring them together and then, really compare apples to apples, right? And so in my original talk title, I also threw in artificial and biological neural networks. Because I'll sort of show you that these ideas can be applied in very general settings, and we're thinking about ways in which they can be used to analyze both brains across individuals and across different tasks, and also analyze artificial neural networks as well. All right. So just to sort of, a lot of the work in our lab has really been motivated by this explosion of data sets that have happened really over the past decade or so, and now even longer. So I've been giving this talk for a while, maybe over a decade. We've been, collecting these really advanced new data sets in neuroscience, both acquiring information at very high resolution and also over very large populations of neurons in the brain. And this has created this sort of neural data deluge. And with all of this information, right, we can record from thousands of neurons now, or if we're looking at whole brain recording methods, even more, perhaps at a different resolution. But with all of this information coming from the brain, we're faced with this challenge of really understanding what to do with all of it, right? And so let me just try to, okay, well, that didn't exactly work. Sorry about that. Okay, so, when faced with all of this information and, coming from recordings in the brain, one way that a number of folks have sort of tried to push forward in understanding how to take all of this complex data and really start to interpret it and understand it is through using dimensionality reduction and representation learning techniques. And so why reduce the dimensionality of data? So a lot of the core principles behind dimensionality reduction really came from its origins in compression and denoising. And there's sort of this idea that if I can find a simpler model of my data or of a collection of data, it should allow me to, store it with a smaller number of bits in the terms of compression, or essentially denoise data by removing outliers or other things that sort of don't fit with this low dimensional structure. And then, of course, another driving reason for dimensionality reduction techniques is really to be able to eventually interpret some of the underlying driving forces that might be giving rise to these complex activity patterns or other variants, right, in our data. So what I'll talk about today is really, driven by this idea that when we look at collections of data, right? If we were to view, just to kind of set the stage and make sure that we're on the same page moving forward, because we'll kind of dig into a bit more of the math here. But if I were to represent just a collection of data as a set of features and a number of different samples, where each of those samples, was represented in some d-dimensional space, if I have d features. And this could be, as I'll show later, maybe my features are individual neurons and their firing rates, and then my samples are, how are this population of neurons sort of firing, or what is their activity pattern jointly? And that could be an example of this d-dimensional feature vector if we had d neurons in that example, for instance, right? And so the driving idea, if we think about this from a sort of geometric perspective, is that if I were to take all of these points in this high dimensional space and I were to look at where they sort of live in the space, the idea is that most natural and a lot of structured signals, images, and audio, and in this case, neural activity, actually lives in or tends to live in lower dimensional spaces than the full ambient dimension, right? So the idea is that we don't have points just distributed everywhere, but they might live towards or be well approximated by a low dimensional structure like a subspace, which has been sort of the bread and butter of techniques like principal components analysis or independent components analysis and so on. And then, of course, there are extensions of this sort of idea where you, instead of believing, that your data might live in a linear subspace, we can also then represent data sets or these low dimensional representations in terms of nonlinear structures, which are referred to broadly as manifolds. Right. And then, of course, we can kind of take this to the extreme. And as we've started to see with more and more complex data sets that we're amassing, that most likely, many data sets in terms of their lower dimensional structures are likely to be kind of complex combinations of many manifolds. And especially if we think about in traditional machine learning, having different classes, right? We could think about this as this union of manifolds where different classes are maybe represented by their own low dimensional structures. Okay, so this sort of just sets the stage for this idea that we're going to be interested in first, finding this low dimensional representation of data. And then we get to this question that's driving really the remainder of the talk is, if I have two data sets and I've formed two low dimensional portraits of each of those data sets, let's just say that for simplicity right now, we've assumed that we've gotten a really good representation of the underlying manifolds that comprise both of these data sets. So a question that one might ask is, can I use this sort of insight or this picture of my data to draw inferences about how two different data sets relate to one another, right? And oftentimes we're faced with this question in neuroscience, right, where we might have two different data sets that maybe from the same animal, but across different across the landscape of a disease, right? So in a control or in a disease state, I sort of alluded to this question of, across individual alignment or comparison, which is this question of, how do two brains solve a similar task, right? Or encode information in a similar way. And then we can also, of course, think about this in the context of, time or across trials or across learning or other comparative settings like that. And I'm just showing a simplified example here, right, where we might have two manifolds that actually have the same shape, but they might have some transformation between them. And, I said at the beginning that we can apply these ideas broadly to both brains and also artificial neural networks. So the sort of analogs of these would be an artificial neural network. If I have a representation that's formed from data, can I compare across different perturbations to the data or different noise? If I have two different networks that have been trained on a task, can I ask questions about how their latent representations are related? And similarly across learning. So that hopefully motivates the reason why we're interested in this question of alignment, or of figuring out how two latent spaces are related to each other. So this is just to sort of describe our approach at a high level. The thing that I will say, which is illustrated on the bottom here, I forgot to have my pointer, is the following. So obviously, if we know that if this is one manifold, right, and this is another, if we know and we have labels on our data that tell us that these points are red and these points are blue and these points are red and these points are blue, then it becomes a much easier problem, right? Because we essentially will have correspondence between some blue points and some red points, and then we could just learn either a nonlinear or a linear mapping, right, between these sets of points and their correspondences. But in general, we would like to be able to understand when we can solve this problem in a fully unsupervised manner, or when we don't naturally have correspondences between points. And I'll talk about some examples where this arises. We're particularly interested in sort of free behavior settings within, neural activity monitoring, where, getting labels is both maybe possible at a very coarse level, but having, point by point correspondence across two data sets is very challenging unless you've structured it significantly. So hopefully that motivates things that, this idea that we have these two manifolds, we want to first figure out at a high level the correspondences between points. And then once we have that, we think, we can find an appropriate mapping between the two. So we can codify this with the following objective, which is written out here. So essentially, mu is going to be sample. So this is a distributional or probabilistic approach in some sense. So we have mu is going to be samples from our source distribution. Nu will be samples from our target distribution, which in this setting, I've already assumed that I'm in the low dimensional space. So we have samples from mu and nu. And then our goal in this formulation is to learn a transformation T, right, that allows us to basically transform our source in order to match the target. And so our goal will be able to solve for this transformation between the two manifolds or distributions in order to minimize some divergence measure, which we'll dig into some different choices for that. I don't know, Vince, if things get interactive at some point, but yeah, feel free if anyone has questions or if there's any things to chat about at this point, feel free to interrupt.
Yeah, we can let you know if anybody has a question. We can certainly. Did you want to ask now? We'll let you know, Eva. Feel free to put it in the chat. Otherwise, we do have time at the end when we'll interact a little bit more.
Okay, sounds good. Excellent. All right, so we have, this is our general problem statement of the distribution alignment sort of strategy that we'll take. And so there's a number of pieces that go into this objective function. And as I already said, if we don't have any labels, this problem can be quite hard, especially in high dimensions. And so making certain assumptions depending on the problem can be really helpful in constraining this further. So we'll talk about a few things like which distance we should use, and I'll give you some insights into why we've settled on some of the strategies that we have and how it's helpful for these sorts of problems in the unsupervised settings. Which class of transformation should we consider? So if we're in the low dimensional space, a very natural first thing that we need to build in invariance to is just unitary invariance, right? So if we have something like PCA, it is unique, but if we use non-negative matrix factorization or other techniques, they're only unique up to some rotation, right? And in general, they're non-convex. So oftentimes, even when we have very similar data that generates very similar manifolds, our latent spaces will only be unique up to that rotation. And so that's at least one important class of invariances that we would want to consider for T. And then the third class of questions is really what additional structure can we leverage in our source and target data sets to help us further facilitate this. Okay, so we'll first dig into this question of which distance we should use. And so just to give people some insights, we, have, I think like a natural distance that one might think about would be something like a Euclidean distance, right? Or LP distance, or in terms of distributions, this might look like a KL divergence, right? And sort of the downside of that, especially when dealing with noisy data, is oftentimes when the distributions don't perfectly overlap, then you'll actually consider that to be really divergent, or these distributions to look quite similar. But maybe if you were to sort of wiggle the points around a little bit, they would actually seem to be a lot closer. So this notion of, kind of encoding the geometry or the space into our measure of distance is really nicely captured with this family of distances called like the optimal transport measures. And so what I'm showing here is just a sort of intuition about why optimal transport works, or why it makes sense as a distance measure. So what I'm showing here are three distributions. I have a blue, purple, and red here. And then over here, I'm just showing what would be the distance under like a LP or KL versus this notion of the Wasserstein distance, or an optimal transport measure. And essentially what it allows us to do is consider the fact that the blue and the purple are actually close in space. And so they should be closer to each other than the blue and the red, for instance. And so I'll show you exactly what the formulation of this is. But this is sort of shown here where the LP, because they're non-overlapping, they all look equidistant, or they're all orthogonal to one another. And with the Wasserstein distance, you have that the blue and purple are close because in space they're also close. And so basically we can write out this optimal transport based measure of the distance between two distributions P and Q using the following formulation. So the key things to take away are the following. So what we're looking for is what's called the transport plan P. It's going to be a matrix, which is, if we're considering, let's just consider the case where we've binned up our distribution into N1 and N2 bins, then our P matrix is going to be an N1 by N2 matrix, which is basically telling us how much mass we're going to move from the entry Pij. Let's see if I have it here. Okay, yeah. The entry Pij is going to tell us how much mass will move from the ith bin in the source to the jth bin in the target. So this is essentially going to encode the moving of mass between these two distributions. And then our goal is just to minimize the total cost, or the total energy required to transport mass across all these different bins. And the distance that we give is, in this case, I'm just writing it down as a Euclidean distance here, but essentially, how close these 2 points are within this latent space, or within this distributional space, will tell us how much weight will associated with transport between those two places. So hopefully that all makes sense. I can't see everyone's face, so but yeah, so essentially the goal, right, is to solve this minimization problem to compute the distance between these two distributions. And then this last term here on the right is basically an entropy term that is going to smooth out our transport plan. And what it does is it allows us actually to have a number of speed ups, and there's nice computational benefits associated with it. And
Eva, quick question for me is, So in the top equation there, where does the information from Q come in? I'm not quite seeing that. Is it in C or is it somewhere in?
Yeah, sorry. So yeah, right. So the cost is going to encode, yeah, the notation is perhaps not super clear. So P and Q are the distributions, and then XIs and the XJs are going to be samples from those distributions.
So J is sampled from Q and I is sampled from P.
So in this case, the PIJ is going to map between, yes, exactly, right.
Okay, got it.
Yes.
Thank you.
Yes. Okay. Okay, and then this is just a visualization of what more entropy does. So essentially, if this would be, you know, our source distribution here along the rows and our target is along the columns, maybe this is our original optimal transport plan. So it's telling us, oh, we need to move a lot of mass from here, right, to this point in space. So we see a large value. And as we increase or drive up the entropy, we're essentially going to be smoothing out this transport plan. What's interesting about this is that you can think about it as a form of regularization, just like if you're doing normal least squares and you add in some regularized term, it essentially allows you a little bit of wiggle room in solving this problem exactly. So if you don't have perfect sets of samples that match up, actually introducing entropy allows you to form a better approximation to the transport overall. So it can be seen both as a way of just speeding things up, and you're kind of adding this thing in and it gives you the inexact form, but in many cases with noisy data, that inexact form is actually more general and is a good solution. So this now just sort of demonstrates or highlights the smoothness and some of the nice properties of optimal transport versus, say, the KL divergence. So what I'm doing is a sort of synthetic example where I take this distribution. What I'm showing here is actually, it's real data from a non-human primate making reaches towards different, 5 different targets in this case. Each point is just in 2 dimensions, and so we're just projecting basically the position in X and Y onto the plane, and the colors are the different target directions that they're making a movement towards. Okay, so I take this distribution and then I synthetically rotate it, and then I'm now just computing what is either the optimal transport distance or the Wasserstein or the KL. And so the KL is shown up here on this top curve. In all cases, right, we are getting the global minimum in the correct place, but we can see that KL is just, it's very noisy, and so it can get trapped in many local minima when you're trying to solve for this rotational parameter. In contrast, we can see that, you know, optimal transport with some different amounts of regularization can really allow us to get rid of a lot of those local minima, which in practice is, you know, really advantageous because this is non-convex. Okay, so that's this first part. I hope I've convinced you that using optimal transport both allows us, we have fast and efficient algorithms for it, but it also is in practice, you know, a really valuable tool for avoiding these sort of pitfalls within alignment. So that's sort of, that addresses this first part. And then to get into some of these more the details really of the tools that we've developed, you know, those will take the form of kind of different data sets, and they'll be motivated by specific applications. So then we'll jump into this problem of neural distribution alignment, which I sort of alluded to at the beginning. So the idea here is basically we are, it's similar to the reaching task that I showed in the previous slide. So basically a non-human primate is, you know, instructed to make reaches towards a given target, one of 8 different directions around a center point. And it's a standard center out reaching task. And so they're going to, reach for the target and then come back to the center each time. What I'm showing here over on the right is just the sort of kinematics of this task as the animal is going to, in this case, 4 different targets. And then at the same time as they're doing this, movement task, we're also then recording neural activity from on the order of 100 or so neurons within the primary motor cortex. And really our original sort of idea and goal was to think about ways that we could use distribution alignment to solve the problem of movement decoding directly from neural activity, but do that in a way where we don't assume that we know correspondence or that we have labeled activity or have neural activity and movement at the same time. And so this idea that I described can be sort of used in this context for decoding, and can also give us insights into how we can use this paradigm to then start comparing across different individuals. So in this case, we take, neural activity patterns, we embed them into a lower dimension using different dimensionality reduction techniques. In this case, factor analysis was a robust tool. And then our goal really was in that low dimensional space to be able to see if we could align the neural activity onto this movement target of interest. And so we use this optimal transport approach in this context and develop this new tool, which we call HIWA, or it's a hierarchical Wasserstein alignment. Really the key innovation here is, in addition to using OT and solving the problem as I've described, we're also going to leverage the fact that these data sets actually belong to a sort of mixture of clusters, right? So in many cases, when we have data that is like a Gaussian mixture model or this union of manifolds that I talked before, if we can use that structure in some way, it can really help us to simplify the problem. So that's what this approach does. Essentially, we see here again this idea of this transport plan, but in this hierarchical formulation, we actually solve alignment over all the points as we were before, but we're going to decompose our objective into 2 parts. So in the first part, we're going to solve for this Wasserstein alignment between points in the ith cluster in our source and the jth cluster in our target. So right now we're assuming that we have the cluster labels in hand, but we don't know which clusters match to one another, right? And so we'll find a transformation over points in the ith cluster onto points in the jth cluster. And then what this outer Pij term here is doing is actually figuring out our transportation between clusters. And then this inner term will also have a transport operator that tells us the interplay between individual points within clusters. In this formulation, we basically will solve this for different transformations. So each pair could have a different T, but then we're essentially enforcing consensus in the end. But what's nice is that it allows us to decouple this hard or potentially high dimensional problem, which is normally over the number of points. And now we're going to be able to solve it essentially over these pairwise interactions between clusters, asking for consensus, and then making sure that the algorithm sort of converges overall. And so, yeah, it reduces the computational complexity. And really the contribution, the key contribution of this work was this hierarchical formulation, which can be used as a way of comparing 2 collections of distributions that are comprised of this sort of mixture model. This distributed solver for being able to speed things up, and then also being able to show that we can use this in the context of neural distribution alignment to improve upon earlier results. This is just showing on synthetic data that our algorithm works. I won't go into all the details here, except for to say that, so our method HIWA and HIWA SSC, this is when we're estimating the clusters directly from the data. And this is when we assume that we have the cluster labels, but we don't know all the correspondences. And so we see in some cases, we can do a very good job of estimating and essentially making this fully unsupervised. And then in other cases, we'll see some gap between the two where maybe misestimating clusters might decrease performance. But overall, we found that it's relatively robust to the exact cluster ID. You can have errors and still the method can find a good alignment.
Did you compare it in that plot with KL divergence as one of those acronyms there, or have you ever done that?
Right. So I don't have that here. So in the case of neural data, we had previously published a paper here during my postdoc where we did this using KL divergence. And so we do compare in the paper between KL divergence and the Wasserstein alignment. Sorry. But in this plot, we're using what are more like state of the art or sort of competitive methods for this task, just in domain adaptation and transfer learning in general. And so a lot of them do use Wasserstein alignment. I actually forget, but I think maybe some of these will use a KL divergence, but I'd have to look. But yeah, just in general, the issue is this problem of getting trapped in these local minima. And so, oftentimes KL won't converge to a good solution in that case. Was there another question?
Yes, so Roger Silva is asking, would the divergence comparison plot for OT still look smooth if gamma were 0?
Oh, okay. So where are we here? So it would look smoother than it should look smoother than the KL divergence. I haven't actually looked at that there. It's a good question. But you can see even for these like different variants of gamma, the overall structure is still quite smooth even as we decrease, but that's a good question. I imagine that you're going to, like what you see here is you start to like observe some of these smaller, not saddle points, but it is a local minima here. So yeah, I expect that you're going to see more of that emerge. It's a good question though. All right, so this is now looking at the case where we have this same center outreaching task, but now we're looking at two different animals performing the same task. We have this underlying movement target, as I described before, just from one of the individuals, right? So this is collected at a different time point. So these are, disconnected in that sense, but it is a prior underlying distribution. We get this neural data, and then after solving for this alignment problem, we can get down to this low dimensional and aligned space. And then we can do the same for another individual. And in this case, what's kind of interesting is because we have a shared target, we can basically take another individual's data and map it onto the movement target for the original individual. And so what this allows us to do is essentially kind of go between brains, right? Because now with two different data sets for which we have no known correspondence, by using this intermediate target, we can essentially go back and figure out when a second individual was in the same part of this underlying latent space. And through alignment, we can be sure that, the same points are the same points for both individuals in that case. And so here we're just showing the nearest neighbor accuracy, or in this case, like an error, which is telling us after alignment, so post alignment, how well we do at just decoding the target direction in these cases. And we can do the same for the other individual. And we can see, of course, here, because our low dimensional embeddings are, perhaps overly restricted, we're losing some information. But with that being said, the overall structure of these data has been appropriately aligned in this case. We've also started to apply this now to free behaving animals as well. And so this is just an example where we're using alignment across different across mice over time. And here I'm just showing examples where we can look at the ability to decode either whether or not the animal was in a wake, non-REM, or REM period after solving this alignment. So in this case, you could think about it as a way of doing domain adaptation or transfer. So if you have some decoder or results that are trained, right, where you know information about sleep versus not being in sleep stages, then even as the information of the neural population is sort of diverging over time, we can bring it back and still transfer the labels that we had from those previously labeled data sets. And here again, just showing that we can effectively solve this problem on different types of data sets. Okay, and so what I've talked about up until now is a sort of, really a tool for being able to do this sort of structured alignment, right? Where we're leveraging the fact that we know that the data sets have some clusters or some clustered structure, and then either estimating that a priori and then feeding that into the algorithm, right? Or we assume that we know it and we can use that in the hierarchical sort of alignment strategy. And so then more recently, we've been sort of pushing further on this idea, but now our goal is to really be able to both simultaneously find the clusters along with figuring out how everything needs to be aligned. So solving kind of both of these problems in an end-to-end manner. And so we've recently developed a method which we call latent optimal transport, which I have, I guess, a little bit of time to describe. But I don't have tons of slides, so I'll do my best. Okay, so what I'm showing here is just an example just to kind of illustrate the idea. But essentially we have a source distribution, which is blue. We have a target distribution, which we're showing in red here. It does actually have, the same overall structure, but then there's also these sort of a hierarchical structure that's sort of embedded within this data as well. And so what I'm showing here is just if we think about this transport matrix that I talked about before, right, the P, and then what we're visualizing is for optimal transport, what is the maximum coupling probability between a source point and a target point? And then what we're doing here is just connecting those points with an edge if there's a high coupling strength, right? So this is the type of transport that you would get with an optimal transport. And then there's been a newly introduced tool that we're building upon, which is called factored coupling, which essentially does the following. So instead of mapping points to points, so instead of forming a coupling between every point in your source and your target, instead what you're going to do is basically factorize that probability into a factored form that has some dependence on underlying latent variables, right? And so essentially, as visualized here, if you think about, okay, I have these latent variables, which are essentially like hubs or anchors in this space. If I put down these anchors, which here are visualized as these black dots, now my goal is to figure out where to place these anchors such that I can basically take information from my source, flow through the anchors, and then back out to the target. Right? So if I've done a good job of placing anchors, or like, I don't know, we can think about it as in terms of logistics, putting down distribution centers that are kind of nicely situated so that, everyone has easy access to a nearby distribution center. But then those distribution centers, of course, can transport goods as well between each other and then out to individual people. Okay, and so that's what I'm showing here, this idea that we're going through this intermediate hub and then back out, right? Oops. Okay, so this is a cool idea because immediately what you have is you have a factorization of your transport plan. We can think about this essentially as just forming a low rank factorization if we just have a small number of anchor points, right? But then the problem that emerges is sort of twofold. If you just minimize the overall transport, you can find these situations where your anchors are actually pretty far away from the structure in your data. And then what that does is it makes it harder to interpret what is being transported to what, right? Because you kind of have these intermediate latent factors that aren't necessarily well aligned. So the contribution of this work of latent optimal transport is essentially putting in an additional term of transport. And now what we're going to do is we can basically, in this case, both of these are different formulations of our algorithm lot. But in this case, we're putting down 4 anchors in the source and the target, and then we can put down more in one domain. And so what this allows us to do is essentially start populating both data sets with anchors, and the source and the target have their own anchors. And then we're basically going to form a transport plan between each data set's anchors. Hopefully you can see it here that, in this case, because we have more flexibility in the number of anchors between source and target, we can start to incorporate these hierarchical structures. And it's cool because now, because, for instance, in this example, if we have a really nice mapping between the anchors and source and target, and we know that there's just a one-to-one correspondence, then we have the ability to interpret transport at this higher level as well, which we weren't able to with this technique. So really our goal is to, find the anchors, route the transport, and then build this factorized transport plan. And we're iterating between those steps dynamically. This is showing now just I forgot to put the numbers on here. I'm sorry. This was just an example where we do like a domain adaptation problem just using MNIST. So we have MNIST digits. They're being projected through a latent space of a network here on the left, and the different colors represent the different digits. And then we pass in noisy or corrupted or perturbed images in this sort of domain adaptation setting, or we could put in USPS instead of MNIST, which is a different domain. And then our goal is to really correct for the shift of the domain in this network. And here I'm just showing some visualizations of the results, and I wish I had the numbers, but the results are that we are competitive with other state-of-the-art methods for using optimal transport for domain adaptation and transfer learning. But in addition, we're really excited about this ability to sort of interpret the transport plans more effectively, which is sort of shown here, where we're just looking at this intermediate transport matrix. So I have the P, which I've actually factorized into PX, PZ, and PY. And here in this space, we can very clearly map correspondences across clusters. So that's cool, and we're really excited about how that can be leveraged in the context of neural data sets, as we've shown in the previous slides. Okay, so I've talked about a number of things, and we're making progress towards building in solutions that can, as I said in the previous slide, in this end-to-end manner, be able to find structures in data, align them, and then also give us some interpretable transport plan back. But all of the things that I've talked about thus far have relied on us already having good latent spaces to form these characterizations. And so just this last part is some preliminary work where we're really trying to understand how to do this effectively, both learning representations and also finding out ways to compare them and align them across, say, individuals. And so towards that end, we've been working on essentially neural network solutions that allow us to sort of both form latent representations and also solve these sorts of alignment problems, right? So what I'm just showing here in this next slide, or I can just go ahead and show you, is sort of where we're at. As I showed in previous slides, like when we use, things like factor analysis or other linear dimensionality reduction techniques, we can lose a lot of underlying structure in the data, and that can often, it gives us less information about what's really going on, right? But also it can affect alignment in a negative way. And so we're hoping that through combining kind of neural networks to learn these good latent spaces that can really capture the data more effectively, and also combining this with some of our comparative questions, we can start to build kind of like these, robust comparative spaces to ask questions about how individuals might do similarly at a similar task. And I can go into more details if we have questions here, but essentially the long story short here is that because neural networks are super powerful and this is unsupervised, like just solving this and not putting a lot more constraints on the encoder is not a good strategy. So we've taken to using kind of a multitask approach where we want to both align, but we also need to be able to reconstruct and classify different parts of the neural activity space or the latent space effectively. And if you add in more constraints, then you can get much better representations. And, we're also working on new self-supervised approaches for forming representations within neural networks. And so these are some of the results now, where we're then comparing just what is the representation you're forming with a supervised MLP, right? This MLP can do a very good job of solving the task, but it can completely distort the space. So we're hoping to continue to push in these directions and getting these nice regularized neural network solutions that don't, distort the structure of these manifolds. And then in the last bit of time that we have before questions, I just wanted to quickly talk about some other work that we're doing in interpreting manifolds and representations in neural networks, but this time in the context of neuroanatomy and imaging data. And so this is just kind of more of a teaser slide just to just to say that we've been getting really excited about looking at variability across collections of data and trying to use that as a way of understanding how individuals' brains differ in terms of their structure, and then using that as a way to study disease in the long term, like an Alzheimer's project. So here I'm just showing an example where we've trained a VAE or variational autoencoder model to be able to create images of brains. And this is trained using the mouse connectivity atlas from the Allen Institute. And so really our decoder is able to generate realistic pictures of brain structure and effectively denoise the data, getting rid of some of the artifacts that are often present but aren't systematic and sort of structured in a way where they're seen in the same way across many brains. So this is a recent paper at Mackay. This is, just showing the images we can create. And then really what we were interested in doing, which is kind of related to the topics that I've described now, is this idea of, using interpretability methods as a way of understanding what factors or what features are being encoded within this latent space. And so here we've just created this sort of bidirectional approach where you can both perturb images that's specific to brain regions that you care about and ask how that changes the latent representations in this space. And then on the other side, you can actually go in and make perturbations in the latent space and ask how that's changing different generated outputs. And then throughout all of this, these ideas of manifold alignment and understanding how to compare two representations become super critical, because, as we make changes, we want to see what exactly is being shifted within this underlying latent space. And then, so we can, just as the final piece here, so we can in terms of mapping projective fields within this neural network, we can basically move a latent factor within our latent space. We can look at all the images that are generated at the output of the network. And then here, what I'm showing is just collapsing all those images and looking at pixels that exhibit high variance. And from that, we can basically find specific brain regions that are being highlighted by specific factors within our model, using this sort of perturbational approach. And of course, in neural networks, we can actually move neurons, whereas in real brains, that's harder. But I think a lot of these tools and these ideas can be sort of readily extended across these different areas. So finally, in summary, I motivated this idea of, needing ways to compare and distill high-dimensional neuroscience data sets, or just data sets in general. I talked about this hierarchical OT-based alignment strategy, its application in rodent and non-human primates. Talked about a new approach called latent optimal transport. And then at the end, some work in more deep representation learning and alignment. And just the acknowledgements, so all the folks in the lab that really make all this work possible, shown over, yeah, they're over here on the right. And then our collaborators, we're, thinking about ways that we can really leverage these tools and a lot of the other things that we're doing in the lab across many different model systems. So we're working with collaborators in human and monkey and rodent, and all the way down to C. elegans and worm. And yeah, our funding sources. And then apparently I shifted my thank you slide. Okay, yes, but thank you. And any questions I'd be happy to answer at this point.
Okay, great. Thank you so much. So we have a few questions. I'll just try to read a few of them off for you. One was earlier on when you were talking about the hierarchical formulation. I think there was a question as to if you could elaborate more on that formulation. Jin Lee was saying that they can't see why it's hierarchical.
I see. So it's just a 2-level hierarchy. I'm trying to go backwards. Oh, here we are. Okay, cool. Right. So it's hierarchical because we have 2 transport plans. So the thing that might not be seen is, okay, this is a Wasserstein distance between pairs of clusters, right? But it's individual points within the clusters. So at this level, we're solving for alignment at the level of individual points. And if you notice, this Wasserstein distance here has its own transport plan, right? So we have a inner transport plan, and then we're also doing this transport across the clusters. So there's this sort of inner and outer formulation for what we're transporting. So in that sense, it's hierarchical because we have these 2 levels of abstraction over which we're solving for transport. Mm-hmm. Hopefully that clarifies things. But yeah, you could imagine taking this and kind of making it more hierarchical, right? By adding in more couplings as well. But in this case, it's just it's difference between clusters versus points.
Okay. Another question from Ismail, when you don't have cluster labels, how do you know how many points to place in each candidate cluster? What do you do when the cluster numbers differ between source and target clustering?
Oh, yeah, those are great questions. So what I showed here was an example where we could estimate the clusters, right? Which, you could argue, oh, how do you know how many clusters you have in general? And that's a problem for model selection. And, that's a hard problem, but it's one let's just imagine we could solve it. And when we have estimated clusters, then what I'm showing here is the case where we've estimated the clusters and given those as labels versus if we don't, whether we have the labels a priori, right? So this works in both of those modes. And then the question, though, of what if you have unequal numbers of clusters is an interesting one. It doesn't there's nothing in this formulation that is going to, I mean, if you have mismatch, you can potentially, you know, map multiple, like one cluster to multiple target clusters, right, through this PIJ matrix. But in that case, there's nothing to sort of guarantee that you're going to do anything reasonable. But yeah, I mean, this inner term could be formed over different numbers. But I will say that this formulation here that I talked about after HIWA, so the latent OT, we can more directly deal with this issue. And actually what we're showing here is an unbalanced setting. So where we don't have the same number of digits in the target versus the source. And what we see here is instead of having this nice one-to-one mapping like orange, right, and blue, most of them are, but there are certain targets, right, that aren't seen within the source. And so it's a little unclear what happens in those settings. So essentially in your original transport objective, sorry, when you're solving for this, okay, right, so this is actually a constraint on your transport plan that ensures that it will marginalize to your true distributions mu and nu. And so what happens in more robust formulations is you can basically relax this constraint. Your entropy regularization kind of does a little bit of the relaxation, but there are other forms where you can directly place that into this bottom part here. So hopefully that clarifies. But yeah, no, it's the unequal or unbalanced setting is a very hard one. And it's one I think that is of practical interest. Yeah.
Great, thanks. So I'm going to highlight maybe 2 or 3 more questions, but then Armin, will we be able to transfer the other questions to the Slack channel?
Sure.
Okay, so you generated a lot of questions, Eva, so I think that was a sign of a good talk, but we may not be able to get through all of them, but we'll put them in Slack so that you can kind of go through those and we can kind of have discussions as a group.
Excellent. That would be great. I'd love to.
So we won't lose them, and we want everyone to get their questions answered. So Ishmael has a whole bunch of questions. I'm sampling from yours just a few of your questions now, and we'll get to them in Slack, the rest of them in Slack. But one that I was also related to one I had was, why would you not, in terms of the earlier 2 sections, why wouldn't you have neural activity labels? And my question was related to that, and you got to this a little bit in your 3rd section, but sort of you were always in the latent space, in the first 2 sections, and how are you translating that back to the neural space, right? And for interpretation, et cetera, and what are your strategies there? So.
Right. So I guess like the first part. Sorry, I remember.
It was neural activity labels.
Oh, right. Like, how do you not have that, right?
Right.
Okay. So it depends on what we mean by labels, right? So in this problem, what I'm saying is, okay, if I was trying to learn like an actual BMI decoder of movements, I would need to know the exact position in space that the arm was in for a given time point, and then also have the neural activity, right? So if I want to create a decoder between actual, not just the movement direction, but the physical movement space and the neural activity, then I need some labels where I'm like simultaneously measuring the animal doing the reaching while I'm recording their neural activity. So I would have these 2 matrices and I could just form a mapping, right, between the 2. But so for instance, in like the free behaving setting, we have situations where maybe we know the animal's like sleeping, or we know that they're like drinking from their water bottle, but we don't know these kind of moment by moment measurements of like exact labels of where in this latent space they are. I mean, in the case of movements, it has a physical and tangible kind of interpretation, right? But I agree with you. In some cases, we definitely will know course labels, like, oh, I know that I'm making a reaching movement towards the green, right, or towards the red, or I am sleeping or I'm not sleeping. But if I took two individuals and they're just like going about their day and doing stuff, to know the points at which our brains were kind of like in similar, I don't even know if it's possible for very complicated things, but in this case, knowing when your brain and my brain was, you know, making like an eating movement versus a reaching movement.
It's like watching a movie and trying to assign a category to every second of every movie scene, right? Yeah.
So, but yeah, when you have these higher level labels, you can use them at least to start organizing blocks of the data. And then what we're saying is this kind of more moment by moment alignment would be done within those blocks.
Mm-hmm.
And then the other question was, yeah, going back to the neural activity.
Right. So like you were doing in the imaging, you know, the neuroanatomy example where you're mapping back to pixels and images, for example. So.
Yeah. So I don't know. We could have like different debates about this. I think, for instance, if you were looking at fMRI, it makes a lot of sense, right? Or if you're looking at something where there might be like a localization, right? And so you want to know exactly where in the original input space was, you know, this effect kind of coming from. But in the case, I mean, I think it's also true in the case of individual neurons, but it's not entirely clear to me that each neuron and its identity matters. I mean, if you want to do perturbations or ask questions about cell types or do something more, but if you have a collection of neurons and maybe they're actually quite redundant and they're doing kind of similar things, and you can figure out what they're doing collectively and you have a representation of that, it's not entirely clear that you always want to go back to neurons, but certainly with these sort of approaches, you could imagine, you know, well, obviously with deep learning, it's harder because, yeah, like with the, as you said, with that latter part, you have to go through this nonlinear projector space and understand how things are being impacted. But certainly with PCA, you can go back to individual neurons and say, you know, which neurons were most responsible for giving rise to variance in some direction of the space.
Yeah.
I think it's a good, I mean, it's another set of questions that we're tackling in the lab of like interpretability. And certainly if you wanted to say things about what specific neurons are doing, you would have to go back.
Yeah. I mean, I was just thinking, this is a silly example, but you could get two very distinct clusters of activity, but perhaps those are coming from every other neuron, right? So those kinds of patterns you wouldn't notice unless you actively go back and forth, right, between the data space and the latent space. Well,
and then there's also that those other neurons could be doing something else that we're not recording, right? And in this case, we're sort of biasing ourselves towards picking up on things that are indicative of movements. But yeah, there could be a lot of other things happening in those.
I mean, it's a hard problem in general, but you know, I basically am a strong advocate of going back and forth a lot, right, between those two. Now, let me ask one more question from Rogers, which is, what is a point/sample when comparing across subjects after alignment?
So in this case, we're aligning, so each sample is a time point, right? And so originally it's it's looking at all of the activities of all of the neurons that we've measured from at a given instant in time. And it's usually sort of binned, right? So we have a firing rate and that's our original data. And then when we're looking in these aligned spaces, it's like sort of saying, you know, time point T100 was really similar in its movement contributions to T point, 200 in this other individual, right? So the alignment is actually at a sort of moment by moment level.
Cool. Okay, so we are at 12:04. I'll do one more question, why not? Which is from Mario. can you elaborate on the possibility of aligning multimodal data or multi-organism data?
Multimodal. So I think a lot of these tools can be applied very generally, right? And we can apply them to different data streams quite easily. The question is like, I don't know, I feel like it might just be it's a very hard problem, but I think we'll probably be in settings where we have some partial correspondences, right? Okay, so you definitely need some correspondences between the two modalities, right? Given a similar sample. I know that those correspondences aren't always given, right? You might not have measurements from two modalities of exactly the same sample, but really this idea of finding correspondence, you'll need to have, I don't know, some way of encoding what is similar across domains. And I think actually a lot of these tools could be combined with some partially labeled data. So maybe there's some cool directions along those lines.
Or I guess if you have two types of data collected concurrently, that would be more straightforward, right?
Right, yeah. So if you don't have correspondence between two data sets, but you have both but you know the multimodal correspondence, right? Or you measured them concurrently.
Yeah. Okay, great. Well, thank you so much for your time and your talk. It was really informative and it was fun. So thanks for joining us. And just as a reminder to everyone, we will have a follow-up discussion and content in Slack, and the video will be available as well. Armin, any other comments that we should make? And also just thanks to IEEE, who was a sponsor of this as well. Until next time, and we'll stay in touch. Try to keep these discussions going. Very interesting work.
Yeah, thank you so much. I'm excited to join on the Slack later and chat with people. And thanks again for the invite.
Great.
Good to see you.
Likewise, just across town. Okay. Take care.
All right, bye-bye.
Bye.